In Chapter 4, we're going to look at the mechanism of DNA replication, how errors occur, and how errors are fixed. And then very briefly, we'll talk about some of the ways that mutation affects the DNA sequence overall. So I've included this overview slide. I'm not going to read it. It's here if you would like to pause and read it. It's just sort of a general outline of our chapter from our book publishers. So one of the important things for thinking about how populations change over time is this idea of descent with modification. Now, descent with modification is basically the process of evolution. And it's the original term that Charles Darwin used. Now we tend to use the term natural selection. But when we're looking at this from a genetic perspective, we are looking at descent as continuity of genetic information. Okay? And then over time, that genetic information is modified through errors and selection. And so we can see this when we look at phenotypes of individuals. So Darwin observed that descendants are similar to their parents. So they have similar traits to their parents, just like you have similar traits to your parents, but they are different over time. Now, some of that difference actually comes from having another parent who is not genetically identical to the first one. So there we already see some modification. And then, of course, there is the selection process that works on increasing the traits that are favorable in a population. So a lot of the genetic information is actually going to be similar between the ancestral finch and all of the other finches. And then they are all going to share some amount of information between them because of this continuous genetic information. But because of natural selection, because changes occur in the genome, we end up with all of these individuals who have been modified to fit their environment. This is called adaptation. So if we look at an example of the sort of general population, we can see this idea of diversity with modification. So if we consider the genome as these blue dots, and we consider that sort of the analogy here is that the overall color is blue with some slight variations, meaning that the overall genetic information is similar with some slight variation. So continuity and diversity are balanced because the mechanism of DNA replication actually keeps too much information from changing. It sort of limits some of the errors that happen. And so we have this continuous information. But some changes do happen, and therefore we get diversity. And when we get diversity, some individuals are going to be more fit than others. And so we have variation that evolution can work on. So if we look at these populations, we see this population, we see continuity. They have similar genetic information, but occasionally changes happen. So over time, we might see changes in the population that produces new variants. We have this black variant, a purple variant, and a green variant. Now, each of these is going to have different relative fitness. That means some are going to survive better than others when you look at with respect to particular traits. So these little black dots represent individuals who could not survive. And so that variant is going to die and you're going to lose it from the population. But these other two are individuals with different selective advantages. This one is a trait that allows them to colonize a new niche or a new local environment. And so that is going to increase their fitness because now they can utilize new environmental resources. This other one has a different variation. This variation gives them some resistance to a common pathogen or resistance to a particular disease. Again, this is going to increase fitness. So now we see some changes, some diversity in the genetic information here. So let's talk about DNA replication, because it is the replication process that is super important for this continuity of genetic information, but it also contributes to the diversity. So really quick review from chapter 3. Recall that we have our sugar phosphate backbone. On each side of our molecule, and this phosphate backbone runs 5' to 3' in an anti-parallel fashion. And then inside we have our nitrogenous bases. And remember, the nitrogenous bases are connected by hydrogen bonds. Remember, we always have thymine binding to adenine. This is a pyrimidine bonding to a purine with 2 hydrogen bonds. And then we have cytosine, our other pyrimidine, bonding to guanine, which is our other purine. And this occurs by 3 hydrogen bonds. Now, this complementary base pairing is what allows us to have a template within our molecule. So built into the structure of DNA, we actually have the template for making new ones, which is really cool. In fact, nucleic acids are the only molecules that can direct their own replication. They encode the directions, meaning they can process the directions, and in both their code and in their physical structure, which is pretty cool. All our other big macromolecules, our carbohydrates, our lipids, our proteins, none of those can direct their own replication. So when Watson and Crick first published their article, they proposed this idea of a template. They sort of quickly mention the idea that this molecule is its template. It is a template for its own replication because of that complementary base pairing. Now, there were 3 general models for how this replication might actually occur. And initially, no one knew how this actually happened, but the 3 major hypotheses for how this replication process occurs were the conservative method, the semi-conservative, and the dispersive method. So in the conservative method, you have your parental molecule, and it is used as a template. I'm going to sort of put a half step in here to show you what happens. It's used as a template for the daughter molecule. So your strands are going to pull apart. They're going to be used as a template for your double helix. Let me draw some base pairs in there. Or for making the other side of your double helix. And then in a second step, those strands will pull apart and you'll get back your parental strand when the parental strands come back together and the newly formed strands come together. And so in this model, you conserve the original parental molecule when you make new daughter strands. In the semi-conservative hypothesis, you basically take out that second step. So you have the original parental band, you pull apart the strands, and you use each one as a template for making another strand. And we don't even need the half step of bringing the parental molecules back together because we just stay this way. And then if you do another round of replication, you have your your original from the first round, and then your new strands being made from those daughter strands. Now, these are still all identical. Don't let the colors confuse you. They're still identical molecules to one another made out of their complementary strands. And then dispersive, not even going to propose how this might actually happen. This one kind of hurts my brain. It would require a whole bunch of steps, I think, with the ultimate idea that you have pieces of your original parental strand incorporated with pieces of the new daughter strand. And so you end up with this crazy complicated molecule. I'm just going to tell you right now that this isn't it. It requires too many steps to get new pieces and incorporate them. I've never seen a good explanation for how this could actually work in practice. But the generally accepted mechanism is the semi-conservative mechanism of replication. And this makes sense because it's the simplest one. It's the one that requires the least steps. So in semi-conservative replication, all strands contain one old and one new strand because your old strands are serving as the templates for your new strand. And so if we look, we have the black line here indicates our original strand, our template, and then the gray one indicates our new one. And if you look at these molecules, you can see that they are identical once the strands are finished, once the synthesis is finished. And you have your complementary base pairing and your anti-parallel nature all in two identical molecules. So this is the original strand is often called the parental strand, and then the newly made strands are called the daughter strands. So let's talk about the experiment that actually proved that semi-conservative replication is what is actually taking place. So this is a fantastically designed experiment by two men named Meselson and Stahl in the late 1950s. This is about 5 years after Watson and Crick proposed the structure of DNA. So I don't want you to get too lost in the details here. I want you to understand the general idea of how this worked. And I think that understanding some of the history is really important because we tend to lose some of these really cool old experiments when we're busy thinking about all kinds of fantastic DNA sequencing techniques and things like that. So let's talk about this one. And the whole point is to look at what DNA molecules are produced and their composition. So in order to figure out their composition, you have to be able to identify the difference between the old and the new daughter strands. So this experiment makes use of using different density nitrogen. Remember, nitrogen is incorporated into the DNA in the nitrogenous bases. And by having different densities of nitrogen, you have different densities of DNA, and therefore a way to separate or measure what molecule you actually have. And so they started this experiment with bacteria growing in a flask in a nutrient medium or a broth that contains all the nutrients they need. And in this nutrient broth, they added a nitrogen isotope called N15 and allowed the bacteria to grow and incorporate that nitrogen into their DNA. So after several generations, all of their DNA contained this N15 nitrogen. In a separate tube, they grew bacteria in N14 medium. And so all of the bacteria there would incorporate that N14 DNA solution. And then they extracted the DNA and loaded it into a centrifuge tube. So a centrifuge is a motor that spins really, really fast. And when it spins, it pulls whatever is inside down towards the bottom. So the heavier things are going to move to the bottom faster, and the lighter things are going to stay in the top. Now they filled it with this cesium chloride density solution. You don't have to worry too much about what that is, but basically it slows down the movement and is of the DNA and is denser toward the bottom. And so the molecule will stop once it meets the density gradient, the correct spot in the density gradient of the tube. So when you spin the N15, you have a heavier molecule that moves down towards the bottom. And when you spin the N14, you have a lighter molecule that stays at the top. And so for this experiment, they started their bacteria in N15 solution until all of them had N15 DNA. And then they changed them to the N14. So the bacteria would incorporate N14 into any new DNA that was made. So if we have our conservative molecule, let's talk about what would likely happen, what kind of results we would get. So if we have our conservative molecule and we we grow it in N15 and we switch it to N14 and we allow one round of replication, we should have our original N15 parental DNA